very good morning to all. It is my opportunity to given and for the presentation of FDP. So actually, this is an FDP for the enrichment of the kind of in drug discovery and development, which was a very good topic, was the created by the college now, Vidyan College of Pharmacy, for all the pharmacy profession here. So maybe some are making the best utilization of this opportunity. And here I am getting an opportunity to make a presentation relatively within bioanalytical approaches on drug discovery and development. Of course, so this particular regarding the, I think one of our faculty, maybe recently they are given a kind of an introduction about they given that. So I worked already in the case of an bioanalytical one and all. Of course, my area of research is on bioanalytical only, and where in this bioanalytical field. So of course, I made in many research and publications also, and it is an opportunity for me to make in a kind of relatively within bioanalytical approaches on the aspect of drug discovery and development. So I will move on to the presentation here. The topic here is a drug discovery and development. The word of drug discovery and development is a very lengthy word, because there is many components are involved here, drug discovery and development, where they are given the opportunity for me to make the presentation for this particular FDP. Here is an bioanalytical approaches in drug discovery and development. Maybe this particular content, maybe it's going to helpful for all the audience who are present here, for the maybe with an interested with an analytical one, or interested with bioanalytical one, or with a background of the pharma here. Okay, and in my presentation, I'm planning to make the presentation on what is meant by bioanalytical approaches, and what is the importance of this one, and how it is important in the case of drug discovery and development bioanalytical approaches, and what are the pre-treatments are possible for the before we are going for the bioanalytical approaches here. And in this particular bioanalytical method development, there is many things are involved. So there is a pharmacokinetics studies are involved, pharmacodynamics will be there, and bioequivalence studies are there, toxicokinetic studies are there, and biomarker analysis, bioanalytical method validation, challenges in the bioanalytical method validations, and there is some of the approaches in the preclinical, and followed by there is the clinical trials, how this bioanalytical approach is going to helpful, and followed by there is how this the particular bioanalytical methods which will be helpful in the case of your the marketing surveillance, and as well as the future directions in the bioanalytical approaches, and emerging technologies in the bioanalytical approaches, and followed by there is a bioanalytic collaboration with the bioanalytic. These are all the content today I brought it. Maybe I hope that it is going to become interesting for you and all, and which is going to create in kind of an interest in the bioanalytical drug discovery approaches for you and all. Maybe it is going, if it is going to enrich your knowledge, so which I am going to present it, so it is going to become very convenient, or it is going to become beneficial for the audience now. Hoping that you are going to understand. So what are my content which I am planning today? Okay, so maybe they are given a kind of the 2 hours time now. So in that 2 hours time, some of the time we are going to plan for some kind of a discussions here, maybe which will plan it as soon after the completion of your presentation. So keep ready or any kind of questions which are relevant to my presentation. So I'm, if possible, if I know that, definitely I will help you, and so I can help you in regarding giving the information which is relevant to the bioanalytical the process or bioanalytical approaches in the drug discovery and development. And here, so before moving on to the bioanalytical approaches in the drug discovery and development, so we should know about what is meant by drug discovery and development. Maybe already some of the speakers in their previous presentation, maybe the yesterday or day for yesterday, some of the speakers, they are already given the information regarding the some of the concept about the drug discovery and development. So we know that the drugs are derived from the different sources there. Maybe in the ancient years, so the drugs are the synthesized or drugs are obtained from the plant originally. So, but nowadays we are finding in some of the drugs, majority, of course, there is a plant origin drugs also under the discovery also there. And even we can observe that there is a drugs are synthetic origin, we are going to find it. And the process of this drug discovery and development is a kind of the tedious process, and it is a very complex one. And it is involves a lot of the materials, and the process is completely and costly because it's taking the lot of time for the to come out a molecule into the market here. Okay, and even after coming into the market, so there is again question is that, so that how it is going to take it to the launch for this new product which is accepted for the social and all. So that is also in kind of an the drawbacks are there here when you are doing a kind of a drug discovery and development for the new drug molecules here. And coming to the drug discovery and development, so which we can broadly classify the drug discovery and development, which are having the three components are there. Maybe you are already aware about the drug discovery and development, in which, so there is a first component will be a kind of in design and synthesis, and there is a preclinical evaluation will be there, and clinical trials are there. So I think this particular diagram, maybe the previous the people are maybe shown it, because without this particular the slide or the content, so there is no any drug discovery will not happen, because these are the this particular 1 second. So this drug discovery here, it isn't maybe this particular slide, maybe everybody is giving an commencement of the drug discovery without this kind of in sequence or process. So we could not able to understand how the drug is going to become available into the market. So it involves the various stages any drug is reaches into the market, which can be broadly classified first drug discovery into two. One is the drug discovery stage, another one is the development stage. So where the drug discovery, it is nowadays we are going for any drug discovery within immediately. We are not going for any kind of a synthesis, and we are not going for the identification of that particular for the biological activity. That is outdated only. Now the very systematic approach is going for the drug discovery now. So we are going for the target drug discovery. So target drug discovery means here we can assume that. So for we are planning for any particular the drug discovery for molecules to the market. So we are planning for what is the particular the disease is there. On that disease, we are going to target the particular the formation of the molecules. So that is going to give a better idea instead of making and synthesizing a molecules and identifying its the applications and all. So hence there is the target discovery is very important. Accordingly, there is we are going to make the selection of the particular the disease, and followed by accordingly there is the models are available. So maybe the computer-aided drug discovery models, or we are going to call it as an CAD facilities. So these particular computer-aided drug discovery facilities, which will be giving an kind of the support for the entry of the molecules into the to the final stage. So for that, there is a initial stage we are going to make a kind of an identification of the target, and followed by how to validate it. So then once you are identifying that particular the discovery of which particular the disease you are identifying, accordingly we are going to identify the molecules by identifying its designing, either by using computer-aided drug design facilities are available, or by moving on to the virtual screening and assay development and high throughput screening, drug design, and hit to lead. So it's nothing but there is a formation of the molecules at this stage. Okay, so then we are going to come out with which is a better molecule is there, so that which can be identified now, and we are identifying its what are the facts are there, and we are bringing accordingly to the identifying the lead identification here. Then once there is a lead identification is done, then we are going for the lead optimization here. In this lead optimization, so of course it is going to reach us almost the molecules now into the stage of preclinicals only, where the molecule is ready now, because where they are identifying by using the drug discovery process, nothing but by using the drug design and narrow down to the molecules of the best molecules, which is going to identify the molecules for the particular diseases, then which can be identified by using the CAD lab facilities. Then later we are moving on to the synthesizing of these particular molecules in the laboratory, which is the important one. So based on the drug discovery approach here. So once the molecules are identified and it is synthesized in the laboratory, then we are going for the ADM prediction hits. So ADM prediction is a very most important here, which is gives an information about what is the drug is get absorbed and distributed and metabolized, and elimination or excretion can be identified by using this particular the preclinical studies only. Usually there is a preclinical study which is going to happen in the case of by using the the small animals only. Maybe it may be rats or mice, or maybe the small animals will be selected. So which will be the drug will be taken into the stage of preclinical only. And in the animal model, so where again we are going to check its efficacy and safety of the molecules, and we are identifying the molecules regarding its safety and efficacy. So once the molecule is safe, if you considered and safety in the animals model, and by conducting its all the parameters, maybe in different ADMA facilities, then it is moving on to the what you call as an there is a clinical development, where in the clinical stage, where there is a now previously there is a molecule is confirmed by using its the activity in the animals. Now they are going to confirm whether the molecules can show its better activity, then it will be taken into the clinical trials, where it will be check the efficacy of these molecules in the human beings only. Okay, so again it is going in a different the phases in the clinical trials. Again, it's a vast stage only. Maybe there is a kind of in phase 1, phase 2, and phase 3 will be carried out, and later it is going for the approval of FDA and EMA, then followed by review of approval and post approval. This is how there is the clinical and preclinical will be carried out. Maybe so this is in the stages of drug discovery, from the molecule which is going to start from the drug design in the laboratory, and to reach us into the market. So it is going to take not in a simple 1 or 2 years. It is taking its own time. Maybe sometimes it may be in 10 to 12 years it may be taking, and later it is going. So where here there is involvement of lot of money also, and lot of facilities also, and requirement of the animal studies and human studies, and finally the drug is going to available for the market. Okay, so where today I am planning in this particular drug discovery, in the drug discovery development. So what is the role of bioanalytical person or bioanalyst? How he is going to helpful for the discovery and development of a molecules? Okay, and as you are already aware that, so any drug molecules which is entered into any particular stage, so it will be initially the drug will be when it is given for any particular drug in the animals, either we are to supposed to take it in the identification of that particular molecules from either from the blood or urine or tissue, or maybe any fecal matter or cerebrospinal fluid. These are all the different kind of the materials will be there through which we are to supposed to extract it. So then there is an bioanalytical approach. The bioanalytical person who is going to first take this particular species or the biological sample for the analysis of the drug here. So that can be taken into where there is a drug means there is a question is available. So that sometimes the drug may be in it may be in kind of in large molecule will be there, or maybe the small molecule will be there. If suppose the small molecule and large molecules, so in that cases usually we are going to call it as in the larger molecules in the kind of in proteins and all. How to analyze the proteins present in the drug, which is how the drug is present in any small molecule or in the molecule which is available in the biological origin. Maybe if it is in the any biological fluid, and which can be taken into the process of, there is the process which is called as either by using your analytical techniques are available, either by using NMR or mass, or maybe some of the different analytical techniques are available where we are going to characterize it. So the question is coming here is what is the type of a molecule it is, whether it is a molecule is in the simple micromolecule is there or macromolecule is there. If it is a simple micromolecule is there, maybe the characterization of that is going to become easy. But when there is a molecule is in larger one, nothing but when the Dalton is in more than the 100 or more than 1000 is there, in such cases the molecule is used only, and our micromolecules are the molecules within less Daltons cannot be so easily analyzed when there is a molecular weight is high. Nothing but if suppose the drug of your interest is belongs to proteins or peptide, in that case, so the analysis of the drug is going to become very difficult task here. So in such cases, we are going for some of the kind of the analytical technique, so which can directly characterize your macromolecules also. And in that cases, maybe we are going to use the hyphenate techniques, which can helpful for the identification and quantification of the drug here. Okay, where there is the involvement of maybe your LCMS, or maybe by using the technique in the LCMS by separation of the molecules, and we are going to identify it. Ultimately, drug it is there in the previously in the biological fluid, and we are taken to a kind of in support of bioanalytical instruments, and later we are going to characterize the molecules by using the hyphenate techniques only. And that is going to take place when there is a molecules are in the form of a metabolites. Okay, the drug and metabolites also equally important in identifying the identification and quantification when it is there in the biological field. And there is a biomarker discovery. This is again important, the concept in the case of biomarker. So biomarker also helpful for the identification of certain disease conditions. So these biomarkers also identified by using a kind of an instruments here. Okay, so in such cases, the bioanalytical approaches, we can do it in a, these are the different segments are there. So the question is coming, why bioanalytical approaches are important? Okay, and when you are observed here, there is a bioanalytical approaches where for a particular drug discovery and development, so when your sample is in the biological sample, to determine the concentration of drug and their metabolite, it is very important. Without which, so we cannot identify what is the amount of drug is available in the particular person who consumed, or whether the drug is going to available in the same formulation, whether it is in the same form only, or whether it is going to change its property. These are all we are supposed to identify by using the bioanalytical approach only. Sometimes we thought that drug, whatever the particular person is consumed, and whether it is remain in the same manner in the human system, sometimes it is not going to become same only. So because of the lot of the enzymes or maybe the effect, due to that reason, your interested molecule will be get converted to some other form is there. In such cases, so there is a drug is available in the different form, or it metabolite maybe activity will be there. Nothing but, so in such cases, metabolite is working as an active molecules for the efficacy or to bring any changes in the body of your interested activity here. So hence, the bioanalytical approach are important, not only whether to identify the drug molecules as well as for the metabolites here. Then here in the case of your bioanalytical approaches, where the researchers are gain an interest in the identification of the drug, how much the drug is available in the blood, so which can be well understood by the process is called the pharmacokinetics here. And in case of your pharmacodynamic also, there is a bioanalytical approach as important. So thereby, how much of drug is absorbed, or how much drug is the sedimented or accumulated in the different body fluids, which can be identified by using the pharmacokinetics discovery only. So nothing but the pharmacokinetics methods are also helpful for the bioanalytical person. So thereby we can able to identify what is the concentration of the drug at different time interval here. And in case of pharmacodynamics, where how much of the drug is accumulated and which can bring them different effect here. Suppose when you are going to give a kind of any anti-diabetic molecules, and impact of that, what is going to happen? Suppose usually we are going to observe the impact of anti-diabetic molecules by observing the glucose level only. So when you are going to measure the glucose level, in turn, we are going to get information that how much effectively the drug is going to work on a particular human beings or who is having a diabetic patient only. So in such that, so how the body is responding for your drug molecules can also be equally important, which can be identified by using the pharmacodynamic properties also. So to understand the pharmacodynamic properties, so there is a bioanalytical methods are very important here. And then followed by, there is some more information which is going to happen here. So generally there is a molecules, there are not only the single molecules, sometimes there is the drugs are given combinedly. There is a formation of drug-drug interactions are possible. So these drug-drug interaction can be well understood. Or if suppose the drug interaction can undergo any adverse drug impacts are there, which you can usually identified by using the bioanalytical methods only. So when you are going to identify the concentration of the drug by using our bioanalytical approaches, so which is going to give the information that whether the drug remains as such, or whether the drug efficacy is going to changes, or whether the drug concentration is reduced, or those are the information which you could able to identify by using the drug-drug interactions here. So it is very clear that there is a bioanalytical approach is very much important in identifying the pharmacokinetic activities, and to know its dynamic properties, and to understand the drug-drug interactions. And when the drug is producing its efficacy, sometimes it may, that if it is and drugs are combined, sometimes it may undergoing the drug-drug interactions are possible. And in such cases, your interested concentration of drug may not be available, and which may leads to some other effect. or adverse effect which can be understood so by using our bioanalytical approach here. And additionally, there is a bioanalytical approaches can also help to reduce the time and cost associated with the drug development. So the bioanalytical person can give or bioanalytical the person can give a lot of information regarding the parameters or methods how we are going to do it. So that particular data, bioanalytical data, so it is most important for the drug discovery and development here. So the time is also equally important, the drug discovery and the cost. So both will be efficiently taken care by using by performing the bioanalytical approaches are important here. Okay. So then after knowing the bioanalytical techniques, there is a question is going to arise: what are the different techniques are there? How these particular techniques can be taken in the identification of drug molecules in the biological fluid. So I'm purely talking about the bioanalytical approaches here or bioanalytical techniques in the drug discovery and development, and where there is a different instruments are involved, because when the drug is available in the biological fluid and identification of the concentration of drug in the biological fluid in any biological origin, any biological fluid, it can be identified by using the different analytical techniques are there. Maybe you are well aware that so some of the technique like your LCMS technique, or maybe the GCMS technique, or maybe the HPLC, or maybe the ELISA and NMR. And these are all the technique where it is going to give the information about the concentration of drug and their metabolites here. Okay. There is a question arises. So which are the best method? Which are the best method technique can be taken? So in such cases, I cannot say that only the HPLC method is the best method for the quantification of the drugs and metabolites here. It is absolutely not that. So because LCMS can give only the molecular weight of the compound, but remaining aspects cannot be identified. Hence, there is a need to identify some other best method in the identification of the compound. In the sense that, so maybe the LCMS technique, or maybe the other technique like maybe in gas chromatography technique. The question is that the difference between here LCMS and GCMS. The difference here is when the sample is in volatile. When the sample is volatile and we can able to do the quantification or identification of the sample in the biological origin is easily. And in such cases, so we have to suppose to depend on the method like GCMS only rather than the LCMS here. Okay. And then followed by, we are observed that there is HPLC techniques also, where these HPLC techniques are a basic technique only, where in which we are going for a kind of a separation, then followed by we are introduced that particular technique into LCMS. So here in the HPLC is going to give a separation of the effective separation, or it may be identification, or maybe the quantification is better in the case of your HPLC. And so where we can able to characterize any unknown molecules are there, which are separated and which can be identified by using our GCMS or LCMS is more ideal here. So these are the different analytical techniques are there. So apart from that, it may be an ELISA technique is there. This is again another important and the best method for the measurement of a particular the specific proteins or maybe the antibodies. So nowadays, so without ELISA technique, so it is very difficult to identify any our interested the proteins and antibodies. So where that. So interested biomarkers, which is in identifying the disease conditions. Sometimes the disease conditions are depend on the biomarker which are present in the biological origin. So which can be very effectively identified by using the ELISA techniques. So that gives the more effective identification of the marker. So thereby we can able to identify the disease condition very well. Okay. Then followed by there is a NMR is the one more technique is there, where NMR is also one of the analytical instrument. It is going to give the information about the molecules and their structure and all, which can be well identified and elucidated by using the NMR here. So NMR is also helpful for the identification of sometime drug protein interactions, and which is going to helpful in the early stage drug discovery research only. Then maybe now, so as we're planned for the bioanalytical approaches in the drug discovery and development. So the sample treatment is then the important the aspect where you are going for the quantification of the drug. The pre-treatment, what I'm going to call it as a pre-treatment. So pre-treatment means, so as we're aware that, So the drug which are available in the market, which can be easily dissolved in a suitable solvent, okay, and we can identify the drug concentration. So by taking a kind of in a particular solvent is enough. But when the similar drug is available in the biological fluid, it is not so easy that recovery of the drug from the biological fluid. So which can be done by taking its own time or by taking its separate special methods are there where we are to supposed to identify, we are to apply accordingly. Then only the drug will be available for your interested state where you can able to identify and quantify. Maybe I hope you understand that this particular stage. So there is a sample treatments are different than the sample treatment which are available for the drug molecules. So one is we are talking about the bioanalytical here. Now the sample are the drug molecules in the biological fluid only. When there is a sample or the drugs are in the biological fluid, so their pre-treatment is required for taken the process. Later it can be available for the quantification of the drug by using any analytical technique. If the suppose the drug is available in the simple any drug formulations and quantification of the drug in the biological quantification of the drug in the simple drug or formulation, it is quite easy. But whereas when the drug is available in the biological fluid, it is taking a difficult time or a difficult task we are supposed to identify. Thereby we can able to extract the drug. So here in the particular process, which we are calling it as an extraction process. This is most important stages in the case of our bioanalytical methods, where we are supposed to identify the different extraction process. Thereby the drug can be extracted. Sometimes the drug is going to, when there is a biological sample means it will be associated generally with the different type of proteins in the blood. So when there is an extraction of the drug from the particular proteins which is associated in the blood, and it is very difficult task here. So which can be done either by using liquid extraction or solid phase extraction or by using the protein precipitation task is there. So by using this is a different extraction method where we are recovering the drug in a better way. Once the drug is extracted, so then the process is going to become easy here. So when the drug is in the biological sample, where we are going for either biofluid samples or maybe the solid biological samples are there. And when there is a biofluid samples, maybe if it is a sample is in the form of a blood or maybe plasma or maybe serum or urine or any other matrices are there, where we are supposed to adopt in the extraction technique. When there is a sample, if it is belongs to animal tissues are there or solid biological samples, or if it is related with the sample collecting from the brain tissues, it is a difficult task is there. So when there is a sample recovery or sample collection is what is the origin is there, depending upon that, we are supposed to go for a different type of extraction techniques are there. It may be a kind of in solid phase micro extraction or still bar, the sorbative extraction methods are there, or liquid micro extraction procedure we are supposed to adopt, or maybe electromembrane extraction is there, or solid phase extraction is there, or molecularly imprinted the polymer extraction techniques are there. These are all the techniques where the recovery of the drug from the biological origin, either maybe any biological origin, it may be blood or saliva or maybe the any tissue fluid which is present, and extracting that by using these are the techniques we are going to adopt. Okay, and when there is a sample is in the tough condition, that when you wanted to collect the sample from the solid biological fluid is there, where we are supposed to adopt a kind of a pressurized liquid extraction or supercritical fluid extraction. Of course, nowadays we are going to give the more importance for the supercritical fluid extraction, where the sample recovery is very effective in the case of supercritical fluid extraction, then followed by microwave assisted extraction or matrix solid phase extraction. These are all the different extraction method will be adopted where the recovery of the sample. Ultimately, now the sample is in the biological origin is there, which can be brought into the original stage. So again, it is not so easy to get the sample into the original stage or into a good form, because it follows a kind of it follows the kind of recovery policies are there. Recovery policies means we are going to follow a kind of some certain US FDA rules are given, where how much the drug is available in the biological fluid, how much the drug is get recovered by using the extraction methodology is also some of the US FDA guidelines are given. By following that US FDA guidelines, so we are going to take it consideration before proceeding for any particular validations here. Okay, so here I am planning to give the more information about your different type of extractions here. like it may be in kind of in, I told, there is the sample is now, it is in the biological system, biological origin. Maybe it is maybe in kind of in sputum is there, or maybe saliva is there, or maybe the blood, or maybe anything is there. So now the drug is in the that biological fluid. Extraction of the drug from the biological fluid into a clear cut compound, or extracting that, it requires different extraction method, would be a liquid extraction. So in this case, liquid extraction, we are going to take a liquid sample, and which is going to be treated with an different. So then we are going to take in usually the polar and non-polar solvent will be selected accordingly. If suppose biological fluid is in a kind of in polar is there, and we are going to take in non-polar solvent like ethyl acetate or dichloromethane, or maybe the methyl tertiary, butyl ethers, or these are all the commonly used organic solvents are there. So these solvents are taken into a kind of a separating funnel is there, and in that separating funnel, our interested biological fluid is transferred here. Then followed by, we are going to add the non-polar mobile phase, or non-polar the solvents are taken into, organic solvent is taken into the separating funnel, and it will be undergoing the liquid extraction here. So when you are adding a liquid extraction, what is going to happen when the sample which is there in the aqueous fluid, now it is moving on to the non-aqueous phase only. When there is a sample is transferred, either from the, if suppose the sample is embedded in the matrix of biological origin of liquid one, and when you are taken in non-polar solvent, so the sample will be get transferred into the non-polar solvent. So it is ultimately the recovery of the sample from the biological tissues into the finest solvent, which is taken as a non-polar solvent only. So if the condition is that, so it will not so easily entered. So condition is that, so the compound of interest is soluble in any one of the solvent which is taken for your extraction, then the particular drug will be get transferred, and you can do the extraction accordingly. So it requires the different methodology, where in which particular solvent can recover your interested sample into a non-polar solvent is important here, in which we have to make different trials, where which is the best solvent can recover your solvents into a non-polar phase, which can be identified, and we have to supposed to separate the two component. If it is the non-polar phase, your sample is entered, in that case we are going to remove there is a high density and low density, we are going to get a liquid here. So usually there is a high density liquid will be sedimented at the bottom, and low density which will be available in the top, and we are separating it. So we are going to identify the amount of this particular drugs, okay, and which is present in the non-polar phase, which can be identified, and it will be tested accordingly. So again, question is going to arise here. So how this process will be taken place? So once there is a separation of the two component, nothing but previously it was in the biological one, or it's in the biological fluid, or it is in the polar, and now it is entered into a non-polar. So now when you are collected the non-polar solvent, where in which you are consider that drug is in the non-polar solvent, in that particular non-polar solvent is separated in the separating funnel, and it is collected in the separate beaker only. Later it can be evaporated drainage, or by using any other treatment where you can able to evaporate the solvent, and recovery of the sample can be done here. This is how there is a liquid extraction can helpful for the separation of the sample, especially the sample belongs to the small molecules in the biological fluid can be taken here. And so I'll move on to the one more important methodology here in the case of extraction of the drug by using the solid phase extraction. As we were observed in the previous liquid extraction, where the sample is insoluble, soluble in the organic solvent, will be transferred into a non-polar solvents, and that will be separated, and it is evaporated, and you are getting in molecules of your interest. Sometimes the liquid methodology may not work in a better way for the separation or the separation of your interested molecules. So in such cases, we are supposed to move on to the methodology which is called as a solid phase extraction, where here in this solid phase extraction, we are going to use the kind of cartridges only. So these particular cartridges, which consist of an stationary phase, interested stationary phase will be there. So this interested stationary phase will be embedded or packed, and this we are call it as a kind of cartridges only. These are all kind of cartridges where in which the stationary phase is embedded. Okay, it is appears like a kind of a small the syringes only, and in these syringes, so the internal part will be in kind of a stationary phase, or kind of inert material, or it may be in kind of in silica, or maybe you are interested what is the type of an stationary phase. Accordingly, there is a type of cartridges are available. So if you are planning to separate your molecules by using the polar stationary phase, or maybe if you are planning for any kind of an following with C8 or C18 or any kind of an, what are the different types of columns are there. In the similar wise, there is a different kind of an cartridge containing stationary phase are available, where in which your interested biological sample is loaded on the head, on the top of the cartridges, and we have to apply a pressure here. Nothing but, so before we are going, before we have taken the sample here, once the sample of your interest from the biological origin, which is dissolved in a suitable solvent, we have to transfer in the beginning. Later, we had to allow it to undergo the percolation. So which can be adapted by using a technique called vacuum. When you are going to apply a vacuum here, okay. So when you are going to apply a vacuum here, there is a separation is going to occurs here, in which your interested molecules are get separated in this stationary phase, and followed by, so once there is a samples are get collected here, so there is a elution will be carried out. The thing is that, so either you are taken the interested molecules of, if you wanted to separate the molecules in your biological origin, it may be retained in your stationary phase, or maybe it may be get eluted. There are two opportunities are there. So if it is settled in the stationary phase only, then you are going to get only the impurities may be trapped, or impurity may be get separated, which are associated with the biological sample only. And in such cases, we have to identify, or we have to examine what is the elution, what is the component which are collected here. So these particular droplets are collected, and we have to examine whether the separated one is a drug one is there, or separated one is an impurity is there. So based on that, we are supposed to examine it, and we have to come to conclusion that the separated one is an impurity or the drug. If you confirm that the separated one is an impurity, so the drug will be there in the your stationary phase only. Again, we have to do the kind of an elution technique, and nothing but, so the solvent in which you are taken is should be a different one, and thereby the drug which is present or embedded, or it is settled in the stationary phase, which can be done by using the elution step here. By doing the elution step, elution protocol, where you can, the drug which is present in the stationary phase, which can be recovered, and by using the elution technique here. So we can collect the fractions now, which are containing in the stationary phase. Nothing but, where there is two things are going to happen here. Either there may be a possibility that your interested drug may be retained in the column, or it may be eluted out. If it is eluted out, so impurity may be stick on into your stationary phase, and the fractions are collected. Then these particular fractions, fractions are identified whether the separated one is impurity or separated one is drug only. So these are all the solid phase extraction procedure which will be carried out in the laboratory for the confirmation of your drug interest. So these protocols are very much essential when your drug is in the biological origin only. Okay, and these methods are called as an SP method, that is solid phase extraction method. So which gives a better accuracy and precision compared to your liquid. So when you come to the accuracy is concerned, so this SP is considered the best technique compared with the LLE. The intention is that, so your LLE can separate your component into one particular polar or non-polar phase only. But whereas here in the case of your solid phase extraction, there is a complete bifurcation of your impurity and as well as your interested components are there. Hence, so your SP methods are considered as an best method compared with your LLE methods only. So then after knowing there is some other methods called, we can call it as so there is a dispersive liquid micro extractions are there. So which is also called as quenchers here, where in the case of quenchers, they are going to make this method ultimately the separation of your interested biological sample in the biological origin can be done by using the liquid micro extraction technique, and which is called as quick, easy, and cheap and effective, rugged and safe, which can be brought in the word of quenchers only. Of course, you are aware that quenchers, but it is not the quenchers, it is a quenchers, where in these quenchers there is a quick, easy, and cheap, effective separation is possible by using the dispersive liquid extraction method only. So which is given diagrammatically in this presentations here, which you are observing that there is a drug which is of your interest. So where here in this particular technique, where the polyaromatic hydrocarbons are separated by using this particular techniques here. Okay, and initially the drug will be mixed with your the extraction solvent here. Then there is a formation of micro extraction, but then we have to do the kind of a centrifugation here. That centrifugation, the separation which separates your hydrocarbons from your polar phase, so thereby it will be get separated and you can able to extract accordingly. So this is considered one of the fast technique where you can able to separate the hydrocarbons. And some more the techniques are there in the case of your bioanalytical technique, that would be in kind of in protein precipitation techniques are there, where in the case of protein precipitation technique is considered as a still more better technique compared with your remaining all technique. In case of your protein precipitation technique, where the drug is treated with the kind of in protein precipitating agent. It may be a kind of in methanol or ethanol, or maybe sometimes we are going to use a acetonitrile only. This particular acetonitrile, when you are treated with the biological samples and which are undergoing the protein precipitated. So this once the sample is get precipitated, nothing but so the protein which are accompanied with the sample is get precipitated, and it will be available as a droplets, available as a residue in the down when you are doing the centrifugation. So the centrifugation is the process which will be applied for the protein precipitation technique, where we can able to separate the samples from the proteins only. One protein precipitation technique is also considered as the best technique for the separation of the drug is concerned. Okay, so here in this particular the slide, which is going to give the information regarding the how the extraction can be done. So as you know that first we are going to do the in kind of a treatment, either by using a suitable treatment, either by drying or by hydrolysis, and by checking its pH adjustment. Then followed by we are going for a kind of an extraction procedure by using analytes, recovering from the sample, and by following its novel methods only. Then removing the interferences. So interferences removing is an important process here to get your interested molecules. That interfering element could be either any protein would be there, or maybe any impurity would be there, so that we can able to separate it by using the cleanup process. Then once the sample is available in the form of a, suppose if we consider in the case of liquid extraction, where the liquid is available in the non-polar phase now, and that particular liquid is get evaporated, you are going to get the evaporation, and evaporation can be done by utilization of the nitrogen stream here, so that you can able to the separate. The intention is that here the nitrogen can support, can helpful if suppose if your samples are any the temperature dependent is there, or the temperature can affect the samples. In such cases, the nitrogen can helpful for the removal of any the water content present in the sample, or if you are interested, so you can dry it effectively by using this nitrogen only. Okay, the reduction of the extraction volume, so that you are getting in the sample of your interested, and which can further go for the analytical technique only. Then so here I'm coming to the, so I hope it is audible. So there is no any the problem at this moment. Everything is okay. So there is no any problem in the.
No, sir.
No problem, sir.
It's right, sir.
Thank you.
Sir,
Thank
please
you
continue,
for
sir.
confirming. Because so sometimes I am in the mood of going for the one slide to the another slide in between. If are not, I thought that everything is going well. So just I'm making the confirmation from your end. So
Everything is fine, sir.
yeah, okay. Thank you for the confirmation. So as we are observed that in the previous information, where you come to know that there are different extraction method are the best methods, which I can call it as an either solid phase extraction is the best method, where we are going for the extracting the drug into an best where without removing all the impurities only. Now I'm coming to the when there is a molecules, if it is a micromolecules are there, so that methodology can be adopted. If suppose the molecules, macromolecules are there, or if suppose the molecular weight is higher, or if suppose your interested molecule is in the form of a protein is there, and how to identify or how to separate your proteins from your biological fluid is a question one. So in such cases, there is a bottom-up sample preparation technique is there. And so in this technique, so where the sample is directly taken into the proteolytic digestion. So the sample is now in the biological origin, okay, and that biological origin containing sample will be undergoing the protein isolation, can be done by using the chromatography technique. So either by using the gel chromatography or 1D, so 1D or 2D or the different techniques are available where the migration of these proteins can be done by using the gel chromatography. So the intention of using here is, so your the macromolecules are can be well identified by using your the previous techniques. But when there is a when you wanted to separate the higher molecular weight sample or the peptides or proteins, so it is an ultimately way to supposed to go for a gel chromatography technique only. So gel chromatography techniques are considered as the best method for the separation and identification of the proteinous samples only. So proteinous samples are there. Usually we are going for the gel chromatography method only. In the gel chromatography method, then it is separated accordingly, then followed by it may made into a kind of a band and identifying your interested protein which is present in the biological origin, and that later which can be converted them into a proteolytic digestion. This is a process in which in gel proteolytic digestion is the process in which you are converting an high molecular weight proteins into an peptides only, where we are going to get an, so with the size will be get reduced here in the peptide mixture. So the here there is a chain length of the protein will be higher in the case of your proteins, and which can be brought into a smaller chain is possible in this process. That process is called in solution proteolytic digestion. So the in solution proteolytic digestion is a process which converts your the longer chain proteins into a shorter chain proteins only, that we can call it as a peptide only. So once there is a sample are available in the form of a peptides, and later so the identification become easy because when there is a molecules are in the higher molecular weight. And once the molecules now it is entered into a fragmented one within short chain, and further we can able to identify directly by using the mass spectrometry or by using the LCMS technique, where it is going to give the molecular weight of the sample, and where so this is how they are going to identify the separation and identification when there is a macromolecules are there, when there is a high molecular weights are there. So this is called as a bottom-up sample preparation technique. Bottom-up means, so they are going from the top, they are going directly taken the sample and they're moving on to the higher, moving on for the separation of that individual constant into a short chain amino acids, and then followed by identification by using the LCMS only. So this is called as a bottom-up sample preparation approach. There is a bottom-up sample approach still can be well understood in this particular sequence only. So previously your sample is in the biological origin only, maybe derived from the cells or tissues or any biofluid is there. By making a kind of in various treatment, we are converting that the samples into a protein mixture, separating them into your interested proteins, then that is converted into a peptide mixture only by following the process called an extraction, or by using a kind of a different process where we are adapting maybe digestion or purification or fractionation. These are all the different techniques are there, and where we are converting your sample into an you are interested peptides by the sequence which is called as a bottom-up approaches only. So here in the case of bottom-up approach, we are converting the sample into a high molecular weight into a low molecular weight in the form of a peptides. Later it will be analyzed by using the LCMS technique only. And there is one more technique is there in the case of your analysis of the high molecular weight by using a top-down technique. So this top-down technique means in the previous technique, as you are observed that the sample will be subjected to the digestion, and followed by the digestion is followed by separating them into peptides and we are analyzed. But whereas in the case of the top-down sample analysis, that is a bottom-top, and whereas here it is a top-down is there. That is that. And here in this top-down approach, that is called top-down sample preparation approach for the analysis of biological by using LCMS. Just observe if suppose if you are taken the sample from the brain tissues, and extraction of this sample is very high task is there, because so here the sample is in the high molecular weight, or if you are interested the sample which can be taken directly, fluid will be collected and it will be analyzed directly into the LCMS here. So it is called as a top-down method. The sample will be directly subjected into an LCMS or maybe the GCMS. then it will be identified. So then once it is identified by using a software called data analysis with some softwares, and we can able to identify what are the different types of peptides are there that will be identified. So in the previous case, so there is a technique where we are adapted bottom up, and whereas here is a top down is there. So in such cases, directly the sample will be subjected for the fragmentation by using LCMS, and the LCMS is going to give in the data. Thereby, we can able to identify what are the different peptides are present in that. So this technique is called top-down the sample preparation technique. So of course, this particular slide, which will give you more idea about top-down approach and bottom-up approach. Observe now, we are discussing about the different bioanalytical approaches here, whether the sample is in low molecular weight or high molecular weight. When there is a sample is a drug molecules, we can go for a simple liquid extraction method, and all we can able to identify, and where you can able to separate easily. When there is a samples are a biological origin, if it is a kind of you're interested, when is a protein is there, and protein can be quantified and separated, because there are different type of proteins are there. What is your interested protein, that segregation, that separation is important here. Either you are going to do a top-down approach or maybe the bottom approach. Already we discussed about the bottom approach and top-down approach, when in which one is directly your biological sample of protein, and without going for a digestion, it is subjected into an LCMS, and fractionation of that into an peptides is analyzed. That is called top-down approach. And whereas in the case of bottom-up approach, where the protein samples is digested, okay, by using a kind of a chemical proteolysis or by using enzymatic proteolysis, where the samples are get converted into a kind of an peptides here, okay, that is called as a proteolytic peptides, and then later it can be subjected into LCMS. So it is very clear that LCMS is one of the best method for the quantification, for the identification of your interested one. Majority of the cases, we are going to for the identification of your interested proteins only. Later, of course, the same instrument can be helpful for the quantification also. So thereby, we are identifying our interested proteins only. Okay, then there is some more methods are available here. So for especially the quantification or for the identification of the samples in the biological fluid, or it may be a kind of an SP method would be there, where in the case of your which I told in the previous one, when there is a samples, if it is in the low molecular weight is there, if it is in nice below the 1000 is there, 1000 molecular weight is there, so which can be adapted by using your LLE method, or maybe SP method, or by using protein precipitation technique, or by using micro extraction technique. So this technique can convert your sample into a very purified form, and once if you got a very purified form, it is eligible for subjected into a next process by using either HPLC or GC or LCMS. Any technique can be adapted, but the purification of the drug is most important, which can be done by using the extraction protocols only. And here, this particular slide is going to give an information about how your sample size is there. So the sample size, and accordingly, there is a time also important, where you are observing that this particular the slide is going to give an idea when there is a small molecules are there, and when your interested molecules are antibodies are there, or oligonucleotides are there, or therapeutic conjugates are there. And x-axis, you are observing that there is a bioanalytical techniques in the x-axis, and in y-axis we made it in structural complexity. When there is a structural complexity is very higher, okay, and so it is taking its own time for the identification and separation, or maybe the confirmation of that particular peptide or the protein is going to become delays, or it is taking its own time only. So hence, it is very clear that if your molecule is the smallest, simpler one, it won't take much time. By using your LCMS, you can able to identify. When there is a molecules are becoming higher one, like the antibodies are there, or maybe any interested proteins are there, or nucleotides are there, RNA, DNA is there, and it is taking long time, and assay procedures are going to become complex here. And another one is in high throughput technique, where we are going for the separating the our interested molecules by following the high throughput that bioanalysis only. where in the case of high throughput bioanalysis technique, where we are going to take in separation and purification can be done by using the directly the evaporating the samples and then followed by subject it into an LCMS, where you can able to identify here. So this is a technique where you can able to identify. So the by using a concentration and drying, ultimately here in this case, the sample will be directly purified, and by using a kind of a nitrogen pressure, then it will be applying a kind of heating in a water bath and taken directly for the analysis without going for any the previous methodology and all. So this is called as a high throughput technique, where in which it is going to characterizing the samples by using the high throughput technique is there. So this is also adapted for the identification and quantification of the drugs here. So now, so we came to the important stages here, that we are in the bioanalytical strategies for the biologics. We are in the virtue of, there are different strategies are involved in the biologics here. Since biological sample is derived from the whole blood, maybe serum or plasma or urine, and when there is an amount of the sample is the small, okay. So the low drug concentration you may get, okay, or many interfering element you are going to get it, and so then differences, there is a expected amount you may not get it. So therefore it is very much essential to establish very sensitive and specific, accurate, the method has to be carried out for the quantification of the drug here, okay. And so here in this case, as you are going to observe that there is an, when you are going for the sample properties, we are supposed to identify their distinctive the properties we are supposed to identify, and based on that, which is the best technique we have to identify whether the molecular weight is lesser or more. Based on that, whether the mass is ideal, or whether you can go for any kind of an ELSA technique, or whether we can go for any chromatography technique or any gel chromatography technique, which is an ideal that should be validated and all for a long time. So this the technique where your gold standard, but where we are going to consider that there is a some of the technique, ELISA techniques are also considered as one of the best technique where for the quantification of the drug here, where the samples will be if it is in the biological origin. The coming here already we discussed about what are the best analytical instrument which can be taken for the quantification here. Either we can take the LCMS or maybe the GCMS and all, it can be taken, or some immunological methods like radioimmunoassays, enzyme immunoassays, fluorescence immunoassays, and microbiological assay methods can be identified if it is in microbiological methods when you are planning, when your samples are in the antibiotics class of drugs are there. So it is very clear that, so there is no only one specific method or only specific instrument which is helpful for the quantification of the drug, okay. And there is some more ligand binding assays also there. So this is again important techniques where ligand binding assays, which can give them better idea, better separation, better identification for the, especially for the large molecules, proteins or peptide by using this. So these ligand binding assays are very sensitive and can measure your protein level as picogram level. So picogram level of your large molecules can be identified by using this ligand binding assays here, okay. And here in this case, so there is a method like your ELISA or electroimmunoassays or automatic, the nano upgrade immunoassays workstation can be used, and where we are going to subject it to a kind of a technique. Okay, and here we are seeing that there is a the stages they are going to give here. Okay, so here in this particular the slide, you are going to observe that how the ligand binding assays can be helpful for the separation and identification. Ultimately, the LCMS is an the technique where it is going to identify the interested proteins and all. Okay. Then there is a under the chromatography method, there are different mass chromatography method or maybe the higher hyphenate techniques are available for the quantification of drugs here. best method can, which I already told, that either top-down approaches or maybe the different techniques where the ultimately we have to identify the proteins which are present in that, if it is macromolecules are there. Okay, either LCMS is considered as the best technique only. Of course, in case of your proteomics, that is for the when you are wanted to analyze the proteins, so the LCMS are the best methods which you are considered right now. Okay, and here there is some more methods are there for the quantification of drug when there is a molecules are higher molecular weight, like hybrid ligand binding assays are there, where again connected to the LCMS only. Hybrid ligand binding assay connected to the LCMS. With the development of complex therapeutic agent and need for the robust analytical tools, so there has been a gradual merging of two technological areas by merging in your the ligand binding assays with LCMS. So this is going to give some more better accurate idea for the separation and as well as for the identification of your interested proteins here. And in this case, they are going to separate by using immunoaffinity isolation. The intention of this particular technique, if suppose there is a biological origin, your sample is in the biological origin, where we are going to take it a technique like the hybrid ligand assays, where the separation of that particular DNA and isolation can be done effectively. And once it is get separated, okay, then later it will be taken for the chromatography for the identification of the DNA and RNA and all. Okay, and here this particular the technique which we can summarize here, that is which is the best methods can be taken, and in the methods which are interested one, what are the advantages are there, what are the disadvantages are there. Suppose when you consider the immunoassays are there, that is microfluidic based assays are there, so those methods are considered as the best and excellent methods for the accuracy and all. And when you are going for some kind of the immune polymerase chain reactions are there, which is the best method again for the quantification of your DNA and RNA, so which can be taken in the increase in the sensitivity and can detect the lower femtomolar concentration. Okay, and maybe these are all again many methods are there which are already repeated here. So hence I am not taken here into more time. So I will move on to the important one is here, the pharmacokinetic studies. As I told already in the beginning, the pharmacokinetics are the important the aspect which is involved in the case of our the bioanalytical approach, the pharmacokinetic studies. The pharmacokinetic studies are the studies which is going to give the information about what is the concentration of the drug in the human beings. So when it is subjected to an given to a particular person, and what is the concentration of drug is available at different time interval, which can be well understood by using these particular studies only. So these studies, which is going to give the vital role for the drug discovery and development. So these studies will determine low drugs are absorbed, are distributed and metabolized, eliminated from the body. This is which can be monitored. So pharmacokinetic studies can be monitored by taking the blood at different time interval, we could able to identify. So to carry out these pharmacokinetic studies, so there is either we are supposed to withdraw the blood after giving a particular dose of a drug to a patient, and we need to withdraw the blood at different time interval. So by understanding this, so the research can better identify its drug efficacy and safety can be obtained. Additionally, this pharmacokinetic studies can provide invaluable information about drug-drug interactions and which can help to prevent adverse drug events. This is most important concept in the case of drug interaction, as when there is a pharmacokinetics data, if it is you are expected the concentration which you are observed at different time interval based on this half-life period and all. So it's considered that the drug is properly absorbing and following its ADME properties are good, and it can be eluted accordingly. If suppose the drug is undergoing any kind of any interactions are there, definitely we are supposed to think in a properly. If suppose expected drug is not available in the kinetic level, the intention is that if the drug is undergoing drug-drug interaction, your expected Cmax and Tmax will not occur properly. So hence, so the bioanalytical methodology are going to become vary when a drug is undergoing the drug-drug interactions. Okay. Even the pharmacokinetic studies can be well understood. So identifying the particular drug, either by using your HPLC or by using the AMS technique can be adopted, and which gives an very accurate results here. Maybe the effective concentration can be determined from the biological origin by using your the chromatography technique, or by using the advanced chromatography technique like by mass associated LCMS are most ideal for the quantification of the drug here. Because we thought that a drug is available in the biological fluid, but the expected concentration, apart from that, if suppose any other metabolites which are produced at different time interval, which can be easily identified in the kinetic studies here. So hence, it is going to give an information about whether the drug is effectively separating at different time interval, or whether the drug is undergoing any delayed response is there. If it is the delayed response, everything can be identified well by using this pharmacokinetic the studies only. And then maybe this particular the slide is going to give a more idea about how the methodology which is adopted in the case of your pharmacokinetic studies here. Maybe where you are observing that the drug will be given to a particular animals here, and we are going to observe the drug at a different time interval here, and we are going to collect the blood at different time interval, and followed by it is going to give the information about the concentration of drug. So that is one aspect. If suppose the drug, if you wanted to identify in the plasma, then it can be, if it is the plasma, if you are identify the sample or the concentration of drug in the brain, or if you wanted to identify the concentration of the drug in the liver, these are all again the process where you can able to identify that how the drug is get distributed, the distribution of the drug and amount of the drug is retained in the liver or in the brain or in the plasma, which can be identified by using this pharmacokinetic studies only. Of course, there are different parts where you can able to identify by using the kinetic studies only. This is an the curve. This is called as the time versus concentration curve, where in which we can able to identify that if suppose in the initially the drug concentration will be low, because as soon as the drug is administered, so in the body it will be very less only. And when you collect the blood at in the beginning time interval, the concentration which will be low only. When the blood is collected at different time interval, maybe by knowing its the concentration, how much is there, by knowing its half-life period, and we can able to identify the concentration of that in the different time interval, maybe 1 hour or 2 hour or 4 hour or 6 hour, or maybe 8 hours, 12 hours or 24 hours. This is how there is a distribution of the drug at different time interval can be identified by using the pharmacokinetic studies. These are all a kind of bioanalytical methods only, where in which we could able to identify the drug level concentration by using the pharmacokinetic studies only. Again, in the pharmacokinetic studies also, there is a requirement of HPLC techniques are there, or maybe the LCMS techniques, which is better is identified once if your drug is already known. In such cases, maybe the HPLC can be taken better, or still if you wanted to find out any kind of a metabolites which are get separated along with your interested molecules, where we are supposed to go for a LCMS technique only. Again, in the LCMS technique, there are different type of techniques are there. Maybe you are aware that there are different analyzers we are going to use it here. Maybe a simple triple quadruple analyzer, or maybe the time of flight analyzer, which is going to give the information differently. Okay, and so that can be taken into the utilization of this kinetic studies for the identification and quantification of the drug, which we are going to get the response here. So I think this is a particular the sequences in which we are able to identify the drug finally available in the sample treatment. So hoping that these are the stages where in which the kinetic process for the identification of the drug. The kinetic, the data of a particular drug, which can be obtained by using the bioanalytical pathway only. So bioanalytical, the technique can be adopted. Again, which we told in the beginning, when the drug is collected from the particular from the animal here, again, we are supposed to directly, we cannot take it into the injection into the HPLC here. We're supposed to adopt a different extraction method only. Either you are interested of the drug is in the, either is present in the plasma or brain or liver or kidney or heart. So these are all the different where, so it is all depending upon how the study is involved, whether you are interested only in the kinetic level or how much drug is available at different time interval, then simple blood is enough. But suppose if you wanted to identify that how much drug is get distributed or accumulated in that different tissues, where we are supposed to adopt the different extraction process, where you can able to identify how there is the kinetic process will be taken place in the case of your bioanalytical approach. Then comes to the pharmacodynamic studies. So the pharmacodynamic studies is also one of the important study, so which is going to give the information about how much drug is absorbed and how much drug is distributed, and what is the response of your body when the drug is given. So pharmacokinetic studies play a crucial role in the drug discovery and development by providing insight into mechanism of action of the drug, whether it is reached. So concentration can be identified by using the pharmacokinetic studies only. But whether it is the intended application has been done or not can be done by using the pharmacokinetic studies only. So this is how we are going to differentiate the identification or effect of that particular drug in the pharmacokinetic studies only. So from this particular the slide, it gives an information that the pharmacokinetic studies can give the information about the EDM, how much drug is absorbed, distributed, metabolized, and elimination can be obtained by using the pharmacokinetic studies only. And so in the pharmacodynamic studies, how much the drug is produced its effect for your intended application. So if you are taken in kind of any kind of drug which is suppose then any kind of malaria drug would be there, or anti-diabetic drug is there, whether the intended application is survived or not. If suppose whether it is given for the purpose of malaria, whether the malaria is cured or not, which can be identified by observing its some particular the methodology only, where the pharmacodynamic the test which is going to give a lot of information here. Okay, so that is the effect of the drug on the animal or on the human being can be well understood in the pharmacodynamic studies here. So this particular study, this study investigate the relationship between the drug concentration and its effect on the body. So helping the researchers understand how drug interacts with its target. The target may be anything here, which is identified, okay, and how it produces therapeutic activity. By studying the pharmacodynamic study of the drug, researchers can optimize its efficacy and safety profile and leading to a better the patient outcome. This is how we are going to helpful these bioanalytical approaches, which is going to give a more important information for the identifying the pharmacodynamic properties here. So these studies are often conducted in vitro and in vivo using variety of techniques, including receptor binding assays or enzyme acting assays, or some of the image acting tactics also there, where you can able to identify. So the samples are collected at the different after the sacrification of the animals. If it is done, then the particular sample will be collected in a different the sample. If it is interested in the liver, or if it is collected in the brain, or some that particular parts will be collected, and you can go for the subjecting its concentration and whether your interested activity has been occurred or not. So which you can able to identify by using this kind of activities only. So by increasing the complexity of the drug targets and need for personalized medicine. So this is how there is a now which the word is here is coming is an personalized medicine. So this personalized medicine is an again important concept where we can consider that. So the interested medicines, depending upon by knowing its the gene level expression. So now it is important for the treatment of the particular diseases and all. So after knowing the concept about your pharmacodynamic, again, this is going to give an idea about how the pharmacodynamic studies are important and how it will be collected and what are the different assays will be calculated, the knowing its cytotoxicity or knowing its it is a cell uptake assays. These are all the different studies will be carried out under the pharmacokinetic studies only. The intention of these particular studies, thereby, so whether the effect of drug is reached and intended application is observed or not, that can be well understood by using the pharmacokinetic studies only. And the another important aspects in the bioanalytical approaches is our bioequivalent studies only. So in the bioequivalent studies also, there is a bioanalytical approaches or bioanalytical methods will be there, where without understanding the bioanalytical, the concept, so bioequivalence studies cannot be carried out. So bioequivalence studies play a crucial role in the drug discovery and development. So determining its interchangeability of different drug formulations. Okay, so these studies are designed to compare the pharmacokinetic properties of the two drug products, such as brand name product or maybe the generic versions of the drug can be calculated. So the results of these bioequivalence studies either by governing by the regulatory agencies are there. Additionally, these studies will help you to ensure the patient receives safety also, and the effective medications can be identified by using this bioequivalence studies only. Then the bioanalysis also involved in the toxicokinetic studies also. The toxicity of a particular drug can be well understood in the case of your drug discovery studies are most important here. So whether your intended drug can produce any adverse effect, which can be well understood in the cellular level, can be well identified by using the bioanalytical approach. So toxicity studies play a critical role in the drug discovery and development by providing an insight into a potential toxicity of a drug only. So by conducting these toxicity studies, so the researchers can identify the potential safety of the drug, and so thereby he can take in kind of important decisions whether the drug does not producing any intended toxicity or possible toxicity can be identified. So for example, if a drug is found to be accumulating a particular organ or tissue, or it may be necessary to adjust the dose of the route of the administration to reduce the toxicity. In that way, we can able to identify the toxicity of the drug also. So under the toxicokinetic studies also, there is an involvement of bioanalytical methodologies are important here. Then comes to the biomarkers. So again, biomarkers gives a lot of information about the disease conditions here, which can be measurable one, can be done by using the bioanalytical methodology only. So biomarkers are measurable indicators that can be used to predict the drug efficacy and safety here, and they can be used to identify the patients who are most likely to benefit from a particular treatment, as well as those who may experience about any adverse reactions. So from the biomarker analysis is an essential component of the drug discovery and development, and it allows research to identify the potential targets and assess the effectiveness of the new treatment. So here in this case, by analyzing the biomarker, researcher can gain an insight into the understanding mechanism of the disease and development more effectively by using the biomarker analysis. So these biomarkers are important here. So thereby we can able to identify the disease condition very well only. Biomarker is not about the drug here. Biomarkers gives the information about the disease condition, and biomarker identification can use the status of disease condition. So biomarker analysis is also one of the important concepts in the drug discovery of the particular interstate molecules. Then bioanalytical method validation. This is another important regulatory aspects where there is a bioanalytical method validations. Bioanalytical method validations are important once the drug is available in the biological origin, and identification of that is important. Identification, which we expressed in the previously, either by using HPLC or GCMS or LCMS or any analytical one. But before taken into the any particular analytical methods, we have to be very specifically taken into the some of the guidelines are there. When we are identifying any particular drug from the biological origin, we are supposed to go according to the validation process are there. So this particular validation process, which is governed by the US FDA procedures are there. By using the US FDA guidelines, they are going to do the identifying the perfect methodology for the bioanalyticals here. So to ensure the bioanalytical methods to use to measure the drug concentration in the biological metrics, whether their method is an accurate, reliable, and reproducible, and important, can be identified by using the bioanalytical method validations only. This validation involves demonstrating the methods meets criteria where there are about around 9 parameters are there, where you can able to identify the different methodology from when there is a sample is in the biological fluid, So as we know that there is a analytical validations, generally we are going for the your ICH guidelines only when there is a sample is in the biological origin. So we cannot take the same the validation parameters here. So bioanalytical method validations are which is taken into consideration from the US FDA guidelines only, and where we are going to take into consideration of the either the bioanalytical method validations like it may be linearity would be or sensitivity or precision, accuracy, stability studies, and specific selectivity and recovery studies are important under the this particular technologies. Where when the particular technique taken for the any quantification, which is follows the these many validation protocols. If it is method is get validated perfectly as per the guidelines which is given, then the particular sample can be taken for the quantification. Okay, this is how we are going in the beginning. Before taken any particular sample, the sample will be get spiked into a suitable media and it is taken accordingly. So once the method is get validated, so again the question is coming here is what particular instrument can be taken for the validation. So there is a LCMS, HPLC methods are also considered as one of the bioanalytical method validation. Or even we can able to take the LCMS method also, we can able to take it, or GCMS method. Either whether which is the instrument we are going to use it, or for that particular methodology, so where method has to be established. So where the sensitivity, it is going to give the information that whether your drug could able to determine by using that instrumental technique or not, whether this particular instrument can able to detect your concentration effectively or how much sensitivity it is there. So that can be taken accordingly, whether GCMS can be taken or LCMS can be taken by knowing its sensitivity only, which is the best instrument is there. That we can consider as under the selectivity, where in the case of selectivity, it is going to give the information that, so which is the best technique instrument can be used for the determination. Based on that, the selectivity and specificity, we are moving on to the different the sensitivity, how much your drug is going to respond, and we are going to establish the linearity of that particular your drug of interest and establishing the linearity confirmation, whether then followed by following its validation protocols in the form of a precision or accuracy, where on spiking the accuracy, where here the recovery of the sample is important, which can be taken into the by following the standard guidelines are there. Okay, then the stability of the particular drug, with whether it is spiked or into the you are interested biological fluid, whether it is can retain for a particular interested period of time, whether it is stable and which can be able to study under this biological method validation and confirm accordingly. These are all the some of the standard guidelines are given under the US FDA, which is taken for the bioanalytical experiments here. And there are some challenges are there in the bioanalytical approaches. Now there is a sample is either in the biological matrices is there, okay, where either in the blood is there or it's in the urine, and so it is difficult to identify the concentration. Okay, there may be some of the interferences which are generally possible for the quantification of the drug here. Okay, and so those interferences may be supporting for the identification separately without affecting any endogenous matter. Then okay. If suppose the endogenous matter or some other factors which are influencing for the quantification of the drug, and these are all again is going to become challenges for the bioanalytical method, bioanalytical analyst. Okay, another challenge is need to validate this analytical method to ensure that they meet the regulatory requirements or not. If suppose you are interested methodology, so it does not comply the standard guidelines, so then method cannot be taken for the further for the any drug discovery and all. So it should follow the regulatory requirement. What is intended purpose is there, whether it is following the intended accuracy, precision, specificity, and sensitivity, everything is followed or not, that has to be taken accordingly. So in addition, there may be differences in the performance of the method between the different laboratories or the instrument. So when the method is carried out by the different instrument, so your methodology remains same only. So if it is different, again, what is going to happen? There is a deviations are going to happen, and due to that reason, your expected methodology cannot be taken for your quantification of the drug or identification of the drug here. So these are all some of the challenges are there in the bioanalytical approaches. Then I can consider that some of the bioanalytical approaches here, which is coming under the drug discovery and studies. Maybe we can consider that drug safety and efficacy before we are going for the clinical trials in the human beings. So preclinical studies are a critical component of the drug discovery and development, and providing the important insight into a drug safety and efficacy before it is taken for the clinical trials in the humans. So bioanalytical approaches plays a key role in the clinical studies, allowing research to measure the drug concentration, especially either by using the pharmacokinetic parameters in the animal model. So this information is used to optimize dosing the regimen and evaluate the potential adverse effects. Then coming to the bioanalytical approaches in the clinical trials. So how the bioanalytical approaches are distributed, which we are given in the clinical trials also, where in the clinical trials also there is a bioanalytical methodologies are involved, and which plays a critical role in the clinical trials also, where the safety and efficacy of the drugs are evaluated in the human beings. And these approaches, which is going to involve the measurement of drug concentration in the biological matrices, such as in the blood or plasma or in the urine. So by analyzing these samples, so researchers can determine how quickly the drug is distributed or metabolized and eliminated can be identified. So in addition, the pharmacokinetic studies also gives the information, and pharmacodynamic studies also gives the information what is the drug effect on that. Okay. Then I'm coming to the marketing strategy, that is post-marketing surveillance. The post-marketing surveillance is a critical phase in the drug development, where safety and efficacy of the drug is monitored by real-world settings. So these approaches can be used to monitor the drug exposure, assess the drug-drug interactions, and detect the adverse drug events, so that maybe have been missed in the drug clinical trials. Okay, and by using the bioanalytical approaches in post-marketing surveillance, the pharmaceutical companies can ensure their drug which are safe and effective for the patient's profile. And emerging technologies in the bioanalytical approach. Emerging technologies, what are the current technologies or emerging technologies are there in the bioanalytical approaches, which can be improvise your technology, or which can improve the existing technology, can made into an easier one by adopting the different trends are available now, right? Trends in the bioanalytical techniques. Either it would be a kind of a 3D printed system would be there, or smart board based sensors are there, or maybe a kind of a multiplex detection would be there, or a kind of a biosensors can be used, or biosensing drug delivery system would be there, or home testing method would be there, or new biomarker discovery would be there. So these are all the new trends is available in the bioanalytical approaches. So one of the most exciting aspects for the bioanalytical approaches in the drug discovery and development is the potential for emerging technologies to reevaluate the particular field here. So there is a improvisation is happened in the analytical approach also. So these technologies include imaging techniques, or by using super resolution microscopy technique, and to visualize the drug distribution effectively and activity at the cellular and subcellular level only. So in that way, we can suppose the new technologies which are going to helpful for the identification and quantification a better way only. Okay. Some other promising technology which you can consider here is an microfluidics would be there, which enables the high throughput for the screening of the drug candidates, or maybe the biosensors which can detect the specific molecules in the real time. Okay, and these emerging technologies have the potential greatly enhances our understanding of the drug mechanism of actions, and improve the drug safety and efficacy, and accelerate the drug discovery and development process. Okay. After knowing how the new trends are there in the bioanalytical approaches, we can think about how this collaboration can make the bioanalytical approaches easy, or effectiveness is possible. The collaboration is the key in the bioanalytical approaches for the drug discovery and development. With the complexity of the biological system, so it is important to have the multidisciplinary teamwork is required, so that we can able to identify the best effective methodology for the quantification and identification of the drug here. So the scientists from the different field, either from the chemistry or biology or pharmacology scientists together, if suppose they work together, can able to establish the result is possible in the bioanalytical approaches. The intention is that there is a need of an interdisciplinary collaboration is required, so that the teamwork can give the better idea. We can come out accordingly for the more efficient workflows and problem solving is possible. So by working together, researcher can develop an innovative approaches so that may have the possible with a single discipline approach when compared with the single discipline approach. So it is a multiple discipline approach is very much essential. So where the sorting out the problem which are coming under the bioanalytical methods here. Ultimately, the collaboration in the bioanalytical approaches can lead to a safer and more effective drugs for the patients only. And the bioanalysis, the future perspectives, where of course we are consider that the LCMS is an best only, but of course there is a not only the LCMS, maybe high resolution mass spec is also one of the best technique where it is going to give them quantitative measurement of the quantity of the drugs, okay, which are use them very sensitive and robust, affordable results are possible for the scientist, which can be used for the quantification of the drug under the by using the technique called HRMS here. So in this respect, the technical advances are needed when it comes to the stationary phase for the LC, and it is hoped that increased interest in the intact protein analysis will stimulate the development of the materials with improved separation of the properties. So here in the case of biological origin or bioanalytical methods, the proteins are only the major interfering elements here. So how we are going to overcome the problem which are associated with the interfering proteins, especially for the micromolecules, are important. If suppose the proteins are get separated and done the quantification well, and we are getting the results very effectively. Similarly, we can supposed to use the user-friendly software solutions for robust method can be adapted for the identification of any complex mass is there, which can be able to identify resolve it accordingly. So suppose there is a protein bioanalysis which can be required, again interdisciplinary science is much successful here. Okay, with that we can able to do the effective separation or identification of the molecules here. Still we have a suffering from some of the challenges in the bioanalysis. The challenges, it would be an maybe more challenges are possible from the past years due to very demanding requirements in terms of method reliability, sensitivity, and the speed of the analysis. Okay, maybe we are consider that LCMS is the best methodology only, and where for the biological proteins, maybe it requiring its own process. Maybe there are several steps will be required for the separation, identification, and purification here. So it requires cleanup steps will be there, and we're supposed to identify the suitable chromatography method for the effective or separation of the component. Okay, and every instrument is having its own constraint and challenges are there. And so when there is a molecules are becoming complex, and determinations are going to become difficult here. And now the structural identification can be done very well by using your either by using NMR or by using LCMS. If it is characterized well, and you can identify the drug very well in this particular bioanalytical methodology. So here I'm coming to the conclusion of my presentations. So as the time I'm reaching almost now, it is 11:30, 11:40 now. The way I'm coming to the end of my presentation, maybe the conclusion here I'm bringing. There is in the bioanalytical methodology, maybe the pharmacokinetic data is important where here is, but there is a pharmacokinetic data, what we are observing here, maybe the concentration of the drug, which is available at different one. These are all the data, maybe reliable, how much we have to reliable here, and suitable analytical techniques are required for finding the concentration. As we are identifying that drug is available in the particular biological origin, and which can be identified effectively by using the pharmacokinetic data here. And bioanalysis emerges as an important, significant throughout the drug discovery and development. And highest impact of bioanalysis measure of the drug and or its metabolites for the pharmacokinetics or toxicokinetics or bioequivalence, and which can able to identify. So here bioanalysis involves all this process, either it may be in kinetic studies will be there or bioequivalence studies and all. As you are aware that there is a around the global bioanalytical testing services market is expected to grow at a compound annual growth rate of 8.6 from 2022 to 2030. So it is going to reach us around $7.65 billion by 2030 is there. So there is a the cost wise rate is also increasing for the bioanalytical testing in the marketing also. Okay. Bioanalytical approaches may play a critical role in the discovery and development, and these approaches will give them various the alarm techniques like kinetic studies or dynamic studies, and all. It's going to be the well idea for us, and where we can able to identify that. So effective bioanalytical methods are possible either by doing it in collaborative way. It is going to be the more effective. The successful bioanalytical methods are possible. So these are all some of the references which I used in my the presentations, and wherever in the presentation also even I shown some of the references literatures where I collected and documented. Where of course we're doing a lot of bioanalytical work in our laboratory for the quantification and identification of the drugs, either by using a kind of an anti-diabetic drugs or maybe kind of cardiovascular drugs. These are all the drugs we are going to identify in our laboratory. And in our laboratory, these are all the bioanalytical methods are very routinely we are doing it. And with the quantification, we are done by using the advanced instrument, by using LCMS or by using the GCMS here. And where we are identifying the concentration of the drug very well. And so without, by mentioning the standard journals which are available currently in the market, are helpful in understanding the mass spectrometry well only. Of course, I'm not taken more detail about how mass is working and all. Everywhere, the ultimate detection of the drug is and mass spec is an ultimate. So, which is the best journal for the current research which is involved, which can be understood by referring these are some standard mass spec journals are there, and thereby you can able to identify that. So all kind of in bioanalysis reports, bioanalytical quantification of the drug from the biological origin, which are reported in the mass spec journals only, either in Journal of Mass Spectrometry or Rapid Communication in the Mass Spectrometry, or Journal of American Society of Mass Spectrometry, or Mass Spectrometry Reviews, or European Journal of Mass Spectrometry. These are all the standard journals where we can able to identify the what are the current research and how the instrument is going to give the possible methodology for the identification of the identification of the drug from the biological origin and all. We could able to identify in this journals only. So maybe I will stop at this moment, and I will give the opportunity for you and all